right, good afternoon. Today we're going to talk about evolution. We're going to talk about how viruses sustain the changes that allow them to explore new hosts. And you know, most of you probably first heard about evolution through Charles Darwin. That's his notebook where he said, this is wonderful, I think, and he drew a phylogenetic tree of how he thought organisms evolved via natural selection. A couple of years ago, I was at a virology meeting, and I saw this tattoo on a virologist, which is Darwin's I think. Isn't that great? She let me take a picture of it. Now, what's this all about with respect to virology? Well, that's a phylogenetic tree which Darwin said represented how organisms are related to ancestral organisms, and we can do the same in virology, as you'll see today. Darwin looked at animals and plants and so forth and used morphologic criteria to say this is related to that because we didn't have genome sequences. But now we have genome sequences, which is the ultimate way of showing organisms are related. So we can make phylogenetic trees with them. And the science of doing this, using the genome sequence to make phylogenetic tree or phylogenies, is called phylogenetics. And these trees, one of which is shown here, they identify the genetic distance by just comparing the sequences. You can compare a gene or the whole genome, and you can identify nearest relatives. So in this tree, we have 10 different viruses whose genomes have been sequenced, and you can run programs on the sequences which will align them and give you a percent identity and so forth, and that's how you make the tree. And you can say that virus 1 and virus 2 are highly related, and they differ by about 5% or so, and they presumably have a common ancestor. And so that's how we make these trees. We compare the differences in the sequences. Each division is a node, so these dots are nodes, which is the common ancestor. So this blue node would be the ancestor of the two viruses on the right. Often we don't have the ancestor, it's implied. So that blue dot is the common ancestor of viruses 1 and 2. And we can go all the way on. We can compare all 10 of these viruses and make common ancestors to specific groups until we get all the way to the left. And that's the root of this tree, which would be the assumed common ancestor of all the viruses here that we see. And again, often we don't have the root organism. It's implied. The problem is that we can only look at viruses that are circulating now. And if we could go back, we could see whether a lot of these are correct or not, but we can't. We have to imply that there is an ancestor. And we can look at differences in the genomes of these viruses and figure out more or less how long they've been around by their mutation rates. So we'll talk about this a little bit later. So that's phylogenetics. The other aspect of what Darwin suggested, evolution by natural selection, we can look at in more modern terms. We have a host protein, or it could be a viral protein, doesn't matter, but it's adapted to its environment. And then the environment changes. It can be a very subtle change or a new host, a major change. And now the host protein is maladapted to the new red environment, so it undergoes adaptive evolution. Variants in the population are selected that are better fit for this new environment. And those mutations arise during replication of the genetic material. All genome replication is error-prone, and that's the raw material for evolution. You need to have mutations in order for evolution to occur. We now know, of course, that you don't just need mutations. You can have genes introduced from other organisms. You can have horizontal gene transfer, which will give you the ability to enter new environments as well. Whenever we have such adaptive evolution, we then have purifying selection, which removes all the mutations that are not suitable for this particular environment. And what ends up, the host protein in this new environment now, it's different from what we started out with, we can identify codons that have been positively selected to be adapted to that environment. And today we're going to see how we can do some of this with viruses and their hosts. So Darwin would have loved viruses. He didn't know anything about them. He lived way before they were discovered or we even knew about them, yet they are the exemplars of natural selection. In particular, RNA viruses, you can follow evolution in real time. You don't have to look at all the different finches on an island somewhere and catalog all of them over years. You can do an experiment in a very short time and see evolution. And so this would be, if I could go back in time and meet Chuck, it would be cool to tell him about viruses, I think. So today we're going to look at two main questions. The overall theme, of course, is viral evolution, the change of a virus population under selection pressure, which is always the case. Even in a cell culture, if we grow viruses in what looks like a simple cell culture, there are selective pressures going on. It doesn't have to be an animal host. And I want to talk a little bit about where viruses came from, just a bit, because we can't really know very much about that, but more likely where they're going. We can make some predictions. Now, virology provides a wonderful window on the mechanisms of evolution, as I said, because viruses reproduce so quickly, and they have huge effects on populations. And of course, you can study evolution in other environments. Island evolution is wonderful to study because, an island is formed and it gets populated with certain life forms. They interact with each other. They undergo selection. That's a very interesting environment to study because it's self-contained. But viruses have other advantages. For example, because they're dependent on host population, as the populations grow, viruses are selected that can infect them. And so we can have new virus population emerging depending on the inhabitants of a region. Viruses can also be selective forces in the evolution of populations. The population has to evolve to adapt to the virus, otherwise it gets wiped out. So it goes both ways. Viruses and their hosts are dependent on each other, and that's how they evolve. And as you'll see later, viruses have had a huge force on the evolution of living populations. Public is constantly confronted with the reality of evolution as it applies to viruses, even if they don't believe in evolution. Why is that? Because we see new viral diseases emerging quite frequently, like AIDS, West Nile virus. Okay, in my lifetime, I have seen AIDS, West Nile virus in the US, hepatitis C virus, Ebola virus, Zika virus. All these have emerged very recently. Of course, influenza virus undergoes antigenic variation and selection on a regular basis. Common cold viruses, they exist as so many different serotypes that you can get infected over and over again. And of course, drug-resistant viruses like HIV we talked about last time. This is viral evolution. So the next time someone tells you, I don't believe in evolution, you can just tell them about these things. You know that new virus that emerged the other day? Well, that's evolution. Maybe part of the problem is that viruses evolve faster than people can understand. Organisms like us that take a year, almost a year for development and birth, takes a long time for us to evolve. Many, many thousands of years for selective forces to be evident, but viruses can be done in one day. And so if you're used to evolution on a big scale, it may be hard to comprehend. Now, the first part of today, I want to talk about the 4 drivers of virus evolution, which include large numbers of progeny. So viruses are unique because they make more progeny than any other organism on Earth. Large numbers of mutants at the same time. And mutation is an important raw material for evolution. Quasispecies effects. It makes no sense to you right now, but we'll hear about what it is today. And finally, selection. The first 3 are the raw materials for selection. We already know, I think, in this course that virus-infected cells make large numbers of progeny. We've encountered that many times, but here's a reminder. 2 viruses we are looking at, which cause blood-borne infections, hepatitis B virus and HIV. The half-life is relatively quick. That means the time when half of the virions or the virus particles lose infectivity, 24 versus 6 hours. Daily turnover, Half of all the viruses in your blood of Hep B turn over in one day, 90% of HIV. And the total production in the blood daily, over 10 to the 11th for hepatitis B, over 10 to the 9th for HIV. These are huge numbers, and that allows for rapid evolution no matter what the environment. You put a drug onto people who are infected with these viruses, likely is that there is already a mutant in the bloodstream resistant, as we showed the other day by looking at the mathematics. And it's not the case just for these viruses, but all other viruses are similar. Replicating viruses produce large numbers of mutant genomes, always. Viruses are always mutating. And you can only evolve if you have mutants, and they are produced during copying of any nucleic acid, including a virus, a bacterium, a fungus, a mouse, a human. Mutations are always made. In fact, our cells, every one of our cells is different from every other cell in our body by many bases, because DNA replication is error-prone. However, the public doesn't get this. Now, I'm going to complain a little bit. Here's an article from 2015 outbreak of Ebola virus in West Africa, which got a lot of attention because it was huge. It was over 25,000 infections, the biggest to date. And here's an article from Time magazine. The Ebola virus is mutating, say scientists. It drives me nuts because viruses are always mutating. It's nothing special going on here or in a common cold in your upper respiratory tract. What they actually mean, the scientists probably mean it's undergoing phenotypic change, right? An amino acid change that makes some difference. But mutation means nucleic acid, not protein. And so this is incorrect. Scientists at a French research institute say Ebola virus has mutated, and they are studying whether it may have become more contagious. So that is something else. It's always mutating, but it sounds like the mutation is something new. Whether it became more contagious or not is a good question. This turned out to be wrong. It didn't become more contagious. And I'll tell you later why it's now too easy to blame mutations when we see viruses changing. I'll come back to that, of course. Now, RNA viruses, as we said last time, their polymerases, like all other polymerases, make errors, but because RNA viruses have no proofreading activity, they have high error frequencies. One misincorporation in 1,000 to 10,000 bases that are polymerized, and this gives you an average error frequency of 10 to the 4, 10 to the 5. So in a 10 kb virus genome, mutation frequency of 1 in 10 to the 4, you get 1 mutation per genome. So as the genome replicates, every time you sustain a mutation. And 10,000 bases is nothing in an infected cell. You can easily make many thousands of virus particles. So you have a large collection of virus mutants. All RNA viruses do this. DNA viruses, the genome replication is not as error prone as we talked about last time. There's a proofreading function. So they have less diversity and they've evolved slower than RNA viruses. And here's an interesting chart which plots genome size on the x-axis in nucleotides versus mutation rate. This is for viruses and one bacterium here. So you can see that the bacteria, the gray circles on the lower right, they have very large genomes compared to all the viruses, and their mutation rate is quite low. And that's not unexpected because they have DNA genomes, but the size also makes a difference. So double-stranded DNA viruses are the open blue circles. They have higher mutation rate than bacteria, but slower than all the other viruses. It's interesting, single-stranded DNA viruses have a higher mutation rate than double-stranded DNA viruses. And people are working on very hard to understand why that might be. And then, of course, we have RNA viruses, retroviruses, double-stranded RNA, single-stranded RNA, they're all clustered in here with the highest mutation rates. And then over there on the upper left, the virus with the highest mutation rate is a viroid, which is an interesting single-stranded circular RNA that infects plants, and it codes for no proteins. And this is interesting because it is replicated by the host cell polymerase. It doesn't encode any of its own. And so why is the mutation rate so high? Maybe because it doesn't encode protein, but we don't really understand that. So RNA viruses clearly higher mutation rate, and genome size can play a difference as well. Now let's talk about the Quasispecies, which I mentioned earlier. What is this? Back in 1978, this paper was published It was called Analysis of an RNA Bacteriophage Population. And I remember this very well. And we quote, a Q beta phage population, that's the name of the phage, is in a dynamic equilibrium with viral mutants arising at a high rate and on one hand being strongly selected against on the other. The genome cannot be described as a defined unique structure, but rather as a weighted average of a large number of different individual sequences. Way ahead of its time, 40 years ago, it's now 41 years, that was last year's number. Not appreciated by most virologists what this meant, but now we understand that all virus populations are not one sequence. They are a dynamic distribution of non-identical but related replicons, and that's what we mean by a quasi-species. Now, this word is not good for many reasons that we don't need to talk about. It's questionable whether species should even be applied to viruses, but they are. Quasi-species implies that because we have this variation, it's not really a species, but that's not right. So the word isn't great, but it's stuck in the literature, and I'm going to use it because the alternatives are not very good. One of the alternatives that's been proposed is mutant swarms. Sounds like a sci-fi movie or something. So I'm going to use quasi-species, even though the evolutionary virologists don't like it. So this is the simplest way I can tell you what a quasi-species is. On the right is the genome sequence of poliovirus, which I did actually in 1981, 7,442 bases. But that's not what the genome actually looks like. It looks like something on the left. Each of these lines is the genome of poliovirus, and the symbols are different mutations at different positions. And they're all different. And that's what a quasi-species is, a mixture of related but not identical replicons. Replicons just meaning the replicating nucleic acid of the virus. And that's for illustrative purposes. There are many more variants than I can show here on this slide. But the point is, each one is pretty much different. And it may be that none of them looks like the sequence on the right here. But when you do sequencing, you get an average of the population. That's why you get a sequence. It doesn't tell you the quasi-species, although now with next generation sequencing methods, we can look at the quasi-species. We can look at all the members of the virus population on the left here. So what is the implication of this for evolution? Well, when a virus infects a cell, it's a mixture of viruses. It's usually more than one. It's initiated by a population of sequences. Not just one sequence is going into a cell. And then there are selective forces inside the cell or inside the host that will make a selection on those different sequences. And what you get out is going to be different from what went in. And those that pass on to a new host, they have to be transmitted. They're under selection of a different sort as well. And so you go in with a mixture, and a certain fraction of that comes out again. So that's why it's important to understand this. You're not infecting with one sequence, you're infecting with a mixture. So I call this the myth of consensus genome sequences. What I did in 1981 is a myth. For any virus, RNA virus, the genome sequences cluster around an average, but every genome can be different. The exact structure, we're not quite sure. It may be that every RNA molecule in that tube of virus that I have is different from the other one. And this sequence that I have may not exist, but it's the average of what was in the tube of virus that I sequenced. I want you to really get that the virus is not a single sequence. It is a mixture of sequences, and therefore selection is bound to be complicated. This is certainly true for RNA viruses. It is probably true for DNA viruses, although less so. The quasi-species are less diverse. And so the quasi-species is produced mainly by polymerase error frequencies, but it also has contributions from recombination. So RNA molecules can recombine when polymerase is copying one and switches to a different RNA molecule, same virus, a different RNA molecule, making it recombinant with sequences from two different parental molecules. These are all polioviruses, for example, but their genomes differ because they're a quasi-species. So recombination can contribute to the diversity, as can reassortment. Viruses with segmented genomes can reassort. The progeny can be mixtures of the parental segments. We talked about this way back in the beginning for influenza viruses. So The quasi-species diversity comes from mutation, recombination, and reassortment. So what happens when you infect a cell or an organism with a quasi-species? How does the selection look? So there is what we call survival of the fittest, the Darwinian concept, of course. And in this quasi-species on the right, a rare genome with a particular mutation may survive a particular selection event. It could be a new host or a new cell that's encountered, or a slightly different temperature, or some extracellular condition that's different. Doesn't matter what it is. There's something slightly different, and maybe it favors one of these mutants. And so that will be selected and predominate in the subsequent populations which grow out. So that's survival of the fittest, because the mutation conferred an ability to withstand the new conditions. We also have survival of the survivors. So you may have a particular mutation on one of these genomes. Maybe the green star selects out that one, but the other mutations are going to go along with it because they're physically linked. And that's what we call survival of the survivors. The linked but unselected mutations get a free ride. And maybe they will persist a long time. Maybe one day they will be selected for as well. When we start with a quasi-species, the product of selection is a new population that shares only the selected mutation. Now, this diversity is in itself selected for. It's really interesting. You may think, well, why don't we get rid of these mutations? Well, of course, you can't, because they're needed for evolution, but people have done the experiment. Now, here's, for example, a poliovirus polymerase, RNA polymerase. We talked about this some time ago. Remember, fingers, thumb, and palm domain. Active site is up here. The red space-filling depiction is the active site. And there is a single mutation, or a single amino acid change at glycine 64 that has been selected for, which reduces the error frequency of the polymerase. It makes it make fewer mistakes, right? So glycine 64 serine is the change, makes fewer mistakes. So that's what I mean, mutations in polymerases that reduce the frequency. These do not have a selective advantage when they're propagated with wild-type virus. So we take a virus with this mutation that makes it make less errors, and we co-infect a cell that makes the normal amount of errors. The what I call the anti-mutator virus doesn't win. So most of the time it loses, in fact. So lower rates are neither advantageous, and they're not selected for in nature. We don't see any of these mutations that cause lower error frequencies arising naturally. And these mutants are often less pathogenic in animal models. So this has been done for viruses, for bacteria of many different sorts. The conclusion is high mutation rates are selected for. Mutation is good for populations, in our case for viral populations, because it allows them to adapt to new environments. And the diversity is contained within a virus population. A few million virus particle has a huge diversity of genome sequences to allow it to rapidly adapt to a new environment. Makes a lot of sense. Instead of having, one genome sequence may not have the mutation the virus needs to reproduce under a certain selection condition, you have all of them right there, or many of them. An important concept related to this idea that mutation is good for you. And by the way, that's why when we look at differences in human genome sequences, we don't call them mutations. We call them polymorphism. Because first of all, who's to say who's mutant and who's wild type, right? They may all be selected for different reasons. And so we call them polymorphisms. What's an important consideration here when we talk about mutation frequency is the error threshold. So as I think I've begun to show you, mutation is a very powerful tool for viruses. It allows viruses to evolve. But as you must guess, you can't have too much mutation and you can't have too little of it. If you go over this error threshold, you lose infectivity. And if you're too far below it, you also lose infectivity. I'm going to show you an experiment that shows you that for RNA viruses, they're evolving very close to their error threshold, and DNA viruses are way below it. So here's an experiment with a DNA virus. We can take a cell and we can infect it with a DNA virus, in the presence of a base analog called 5-azacitidine. So this is a modified form of C, nucleoside C, no phosphates, it's a nucleoside, which is not exactly C, but when it's incorporated as C, it templates as a T. So the polymerase thinks it's a T and adds an A instead of a G. So it's a mutator, it's a base analog that causes mutation. So that's 5-aza-C. So we've incorporated into our cell culture. So the DNA virus is growing in the presence of 5-aza-C, and mutations are being incorporated into the genome, G to A transition. So you get actually, when you do this with DNA viruses, an increase of several orders of magnitude of mutants, of mutations in the genome. And it's fine. The virus can handle that because, as I've said, it is existing far below its error threshold. We would know if we exceeded the error threshold in this experiment if we lost infectivity. When you do this similar experiment with an RNA virus, which I'm going to show you in a moment, the error frequency can only increase 2 to 3-fold, not several orders of magnitude as for the DNA virus, only 2 to 3-fold. It's already got so many mutations in the genome, that's what the quasispecies is, that you just increase those numbers of mutations by 2 to 3-fold, and now you lose infectivity. How do we know this? Here's the experiment. It's done with an antiviral drug called ribavirin. Ribavirin used to be used to treat hepatitis C virus. It's a base analog that gets incorporated into the nucleic acid by the polymerase, and it causes mutations by similar mechanism as aza-C that I just told you. And so here we have a graph where we've done an experiment where we have grown polioviruses in increasing quantities of ribavirin to get more and more mutations incorporated into the genome. And so on the x-axis, we have the number of mutations per genome. So the experiment to grow the virus in the presence of the drug and then sequence the genome, and you can tell how many mutations are present. And then on the y-axis, you have the infectivity of the genomes. And so in the absence of drug, it's about 100%. There are no mutations. That's the baseline. And then we put 2 mutations. We put 1 mutation in all the way up there, and there's not much of an effect. And then we put 2 mutations in, and now we've gone down to 30% of our infectivity, just by 2 mutations per genome. It doesn't have to be in any particular place, just on average. And then 7 mutations, we're almost at 0, and 15 mutations, we've lost all of our infectivity. So you can only increase the number of mutations 2 or 3 times above wild type before you start to lose infectivity. You're exceeding the error threshold. So this is why I say DNA viruses exist way below it. And why do they exist way below it? Because their polymerases don't make a lot of errors. So they have a lot of room to go to reach the error threshold, whereas RNA viruses are right at it, and they can't exceed it. And they're at it because of their error-prone polymerases that don't have proofreading functions. Now here's another example of the importance of the quasispecies. And this is an experiment which is going to show you that if we put a lot of selective pressure on small populations of viruses, we're going to have a problem. We're going to have loss of diversity and lots of mutations. And so in this experiment, we do a plaque assay, and we pick a single plaque. We expand it and grow it on another plate of cell. We plaque it again. We pick a plaque, grow it up, plaque it out again. We pick a plaque, grow it up, plaque it out again. You do this over and over again. You can do it as long as you have patience. But after 20 or 30 cycles of doing this, you get viruses that can't grow very well any longer. They're barely able to grow. They're less fit than the original population. So why is this? It's the same cells, the same temperature, the same agar overlay, the same medium. What's going on? You're not applying any selective force, as far as you can tell. Everything is equivalent. So why do we have hardly any virus replication after just 20 to 30 passages? The answer is because we forced the virus through a genetic bottleneck. A bottleneck by restricting replication to the progeny only found in a single plaque, which may be a few thousand virus particles, as opposed to millions of virus particles if you took the yield of a cell and put it on fresh cells. few thousand progeny viruses derived from a single. So here's a graphic illustration of a bottleneck. You have a parental virus population, but you can only get a few through the bottleneck. You're reducing the population. The bottleneck is the plaque. You get a surviving population which looks very different from the original one. It's restricted to a few thousand founders, and so your next generation is going to be very different. And in fact, if you sequence these populations, you'll see the change in their diversity. Another way to look at this, besides a bottleneck, comes from what's called Muller's ratchet, which was used to explain why sexual reproduction arose. Small asexual populations eventually will accumulate deleterious mutations. That's what a virus is doing. It's asexually reproducing, right? It's got its own genome and it is multiplying it. There's no contribution from other genomes. And this was used to explain why sex occurs, to rescue these deleterious mutations. So what's happening here is our RNA viruses are close to the error threshold. We restrict the population to a few thousand viruses in a plaque, and there's no selection here, but mutations are accumulating in those 1,000 viruses. They're not immune to error-prone replication, but you've only got a small population with those errors. And that's why the fitness is declining, because you don't have enough population diversity to overcome the fitness cost of the existing mutations. It's called Muller's ratchet. Well, Muller was the guy who thought it up, and it's a ratchet because, that's a ratchet. If you know how that works, there's a little metal cog that engages the wheel, and you can't go back. You can only go forward with these ratchets. And so the idea is each mutation lets the gear move forward. It gets fixed into the genome, but you can't go back again. And you have each round of error-prone replication. You take a few thousand viruses, you allow them to replicate in cells, error-prone replication for sure, but it's a restricted population. You're putting more and more mutations into that restricted population, and eventually it peters out. You lose viability, and you have hardly any virus titer anymore. The environment hasn't changed at all. What's changed is that at each round, you're introducing more and more mutations into the genome, but the population isn't diverse enough to absorb them. And that's another reason showing that you need a quasispecies in order for viruses to be viable. So that's illustrated here. We have on the left large population passages. In other words, not a plaque, but just taking the supernatant of an infected cell and putting several million virus particles onto a new cell culture. You have a whole collection of sequences, the quasispecies there. The population remains diverse. Now, in a plaque assay, we're picking viruses derived from a single founder, so they may likely have very, very little quasispecies diversity. And so here we're taking this particular genome out by plaque assay, and now these original mutations are present in all the progeny that are replicating after we pick the plaque, and we're getting other mutations as well, but because we don't have the population diversity as we do on the population on the left, the fitness of this declines because there's not enough sequence to rescue the mutations that have been introduced. Again, it's a nice experiment which shows why a quasispecies is important. It allows diversity so that deleterious situations can be dealt with. Here is a table showing you this experiment with different viruses, so it's not just theoretical. This is fitness decline compared to the first virus after passage through a bottleneck, a bacteriophage, 40 passages, 22% decline in fitness, VSV, 20 passages, 18%, foot and mouth disease virus, a coronavirus, 30 passages, etc. HIV, after 15 passages, has lost 94% of its infectivity. So this is a real phenomenon that can be reduced with many viruses. So you may ask, is there such a thing as a bottleneck in the real world? Because I've told you this as a plaque assay experiment, picking plaques. And yeah, there are several situations where small amounts of virus particles are the inoculum for an infection. So aerosol transmission, you get infected with an aerosol, maybe a single droplet with a few thousand virus particles in, that's a bottleneck. Activation of latent viruses from small numbers of cells, maybe ganglia, reactivating herpes simplex viruses. Insects, they don't deliver a lot of virus, and so it's a small inoculum, a restricted population. So these are all situations where you could have a bottleneck occurring. Yet, it doesn't happen. How do infections that spread by these routes escape Muller's ratchet? To avoid the ratchet in cell culture, the experiment that we did, instead of picking a single plaque, you could pick several and mix them together and pass them, and then you would no longer be subjected to Muller's ratchet. You've made a more diverse replicating population. This gives you the ability to rescue any deleterious mutations by recombination or reassortment, for example. So you may have a particular deleterious mutation in the genome, but you've got the genetic diversity now to rescue it. And that's shown here by this slide, by reassortment or recombination. This is recombination being illustrated. Let's say we have two viruses, 1 and 2 in our population that we've subjected to a bottleneck. We have mutations that are deleterious, but if they recombine, they can give rise to a healthy recombinant. And having the parental viruses that can recombine to do this will only happen if we have a diverse, large population size which has not been subjected to a bottleneck. And so in vivo, we think these kinds of scenarios of recombination or reassortment can rescue the bottleneck imposed, say, by a mosquito giving you a small number of particles. Diversity of a population is important for the survival of all of them. The entire population survives when its diversity is great. You remove that diversity, which we did by the bottleneck passage, plaque-to-plaque passage, and reproduction suffers because the population doesn't have the diversity it needs to deal with genomes that are accumulating mutations. We can also see evidence of the effect of a quasi-species in the real world, and let's look at a couple of examples. One that I like because it pits the virus against your immune response is the selection called genetic shift and drift. And we've touched on this before with influenza virus, but let me remind you, we get infected with viruses, we make immune responses against them, and it's only natural that the viruses are going to be selected for that escape antibodies or cellular responses. We've talked about this, how changes in epitopes on viral glycoproteins can lead to escape of antibody neutralization. CTL epitopes can change and avoid killing by cytotoxic T lymphocytes. So this happens in an infected individual, an immunocompetent individual. You'll definitely select virus mutants. This is nothing more than selection. The viruses undergo mutation, and you can select out viruses that are not neutralized, for example. And these two kinds of selection, drift is diversity from copying errors in immune selection. So in terms of the hemagglutinin of influenza virus, single amino acid changes that escape antibody neutralization. Shift is diversity that arises after recombination or reassortment. Remember, influenza viruses are viruses with segmented RNA genomes. On the surface of the particle are the hemagglutinin and the neuraminidase glycoproteins. And in fact, this is how we classify influenza viruses, by combinations of the H and N. They're called H, X, and Y. There happen to be 18 different hemagglutinins that we know out there and 11 different neuraminidase. And only the H1, H2, and H3 hemagglutinins can infect people. So if you look at the vaccines that are made, they will only have these components in them. All of these viruses infect birds and other animals. And we already talked about antigenic drift. This is the viral hemagglutinin protein on the right. And when you make antibodies against it, the majority of the antibodies are directed against the head of the hemagglutinin, which contains the receptor binding site. As you make antibodies and you're infected with viruses, the viral diversity is so great that you select for variants that can escape neutralization with the antibody. And these colored sites are epitopes. They're sequences recognized by individual antibodies. A single amino acid change in each of these epitopes is often enough to prevent the antibody from binding and neutralizing infectivity. And so that's antigenic drift. The virus changes in every host, and at one point, a virus will emerge that evades the antibodies being produced in that host. That virus will then spread to another individual and spread throughout the population. And this is one reason why we often have to change the influenza vaccine from year to year, because of this kind of antigenic drift. Antigenic shift is caused by reassortment or recombination, but we're going to illustrate it with influenza virus reassortment. On the left is a depiction of the emergence of some of the pandemic influenza viruses of the last 100 years or so. So back in 1918, had a huge pandemic, a virus that infected the world, hundreds of millions of people infected. It was called Spanish flu. We think that this virus came from a bird of some sort, a water bird, to infect people. And so here's the presumed precursor of this virus. It was a duck virus. 8 RNA segments are shown red here. And that's a virus that gained the ability to infect people. And since no one had immunity to it, it spread globally. That virus persisted for many years. 1957, a new pandemic strain replaced it, called the H2N2 strain. It's called Asian because it first emerged in China and then spread around the world again and infected many, many people. This virus has a number of RNA segments from the 1918 virus, but 3 from a different duck virus, a different duck influenza virus. So reassortment in some animal somewhere produced that virus, which when it got into people was enough to replicate. And since no one had immunity to the H2 hemagglutinin, had never been seen, or at least in recent history hadn't been seen, it infected much of the world. And finally in 1968, similar reassortment leading to the H3N2 virus. You can see now this virus has segments from the 1918 virus, the 1957 virus, and another duck virus, influenza virus. And the most recent pandemic was 2009, when an H1N1 virus emerged. And look at the genome of this virus. It's got RNA segments, so we could trace them all the way back. The purple one comes from avian influenza viruses circulating in 1990. The red comes from human H3N2 viruses that had been around since 1968. The dark blue comes from a swine H1N1 virus. The cyan comes from a Eurasian swine influenza virus. And these segments went through intermediates in North American swine, as you can see here. So multiple reassortment events. This is always happening in wild animals infected with influenza. And so this one was a reassortant with segments deriving from many different animal influenza viruses. And so this is genetic shift. Entire new segments coming into the virus, often presenting a hemagglutinin that we haven't seen in a long time. Now, this H1N1 was around in 1918, but it's in the intervening time, it's changed in humans quite a bit. But in pigs, that H1N1, in fact, in 1918, at the same time the virus infected humans, it infected pigs. And in pigs, because they don't live a very long time, there's not much evolution. And so that H1 segment in the 2009 virus was hiding in pigs all these years. And so enough people hadn't seen it that they could be reinfected. Let's now talk a little bit about how we can identify other host proteins that antagonize virus replication. I've talked about antibodies in terms of drift and shift of influenza virus. It's conceptually very easy to figure out, but there are lots of other proteins in the cell that block virus replication, all those interferon-induced genes. We can actually look at the evolution of these genes in their hosts and figure out how they block virus infectivity. So this is what we call a host virus arms race. Because let's see, we have on the top, we have some host protein that can block virus infectivity. This looks like an antibody, but it doesn't have to be an antibody. It could be something else. And so for some period in evolution, the host is winning it's blocking replication. But you know, viruses can mutate rapidly, so a virus will certainly emerge with a little bump that prevents it from being blocked by this protein. So for a period, the virus will win. But then, of course, the host can mutate to accommodate the bump, and this goes back and forth. And over thousands of years, if we look at phylogenetic trees, we can see evidence of this, as I'll tell you in a moment. So that's what I mean by a host virus arms race. Nobody wins. It's kind of like the Red Queen. Around here, it takes all the running you can do just to stay in the same place. In fact, these are now called Red Queen conflicts because of Lewis Carroll's little statement there. How can we use this to figure out what's going on? Let me show you. You can make a phylogenetic tree. Here we're looking at primates. Okay, all the primates we know, whose genome sequences we now have, we can make a beautiful phylogeny and know exactly all the nodes on that tree. And then we can look in orthologs, which are related proteins in all of these different primates. And we can look for signs of positive selection. That is, a codon which seems to be maintained in a particular lineage. So for here, for example, the red are some evidences of positive selection. And you can see some lineages, the red mutation popped up early and stayed through. That's positively selection. It had some advantage to the host, and so it's maintained. And a key concept here, you can look at these sequences, and there are computer programs that will analyze them for synonymous and non-synonymous changes. As you know, the genetic code is redundant, and so you could make a number of changes in the codon which wouldn't change the amino acid. Those are synonymous changes. The non-synonymous changes result in an amino acid change. And that's what you look for, a high ratio of non-synonymous to synonymous mutations. And you can identify positively selected codons. And so here you can see at this particular position, there are certain codons that seem to predominate. So that's your first start. You take an animal that you're interested in, it doesn't have to be a primate, it could be anything, and then look for the evidence of selection in a particular protein. And the idea is that this could be a virus that's applying the positive selection. If this protein is interacting with a virus, this could be evidence of virus pressure. And then you do some experiments. You take cells from each of your primates, and hopefully you have cells in culture that you can use, and you infect them with your virus. And you can ask, in these phylogenies where there was no codon positively selected, the host seems to win. And when there was a positively selected codon, the virus wins. You actually do these experiments in cell culture, and you ask, does the virus win or does the host win in terms of replication? So when the virus wins, there's a mutation in a particular viral protein that evades the host protein. The host winning, there's a compensatory mutation that can overcome the virus mutation. So you can sort all this out in cell culture, and you can look at the amino acids in the host protein and in the virus protein that mediate this effect. And then you can map the changes on the interaction. If you happen to know how the virus and the host proteins interact, you can have crystal structures of these. You can map these changes. Here's a positively selected codon in a host protein. When it's an S, the virus wins. When it's a G, the host wins. And if you can make mutations at these specific places in the proteins and duplicate the finding at the level of individual proteins. Using this approach that I've told you, a lot of examples of cell proteins whose overall makeup has been completely driven by virus selection. And here are two examples on this slide. On the left is a protein called TRIM5A, which is a cellular defense protein that defends against HIV infection. And the experiments I've just showed you looking for signs of positive selection were done. And here on the TRIM5 alpha are the positively selected amino acids in red. And on the bottom, that's where the HIV capsid binds to TRIM5 alpha. So the TRIM5 binds the capsid, prevents it from infecting, and the virus can evade that by mutation, and then the cell protein can change back. And we can look at the phylogeny of primates over thousands of years and watch this going back and forth to host winning, virus winning, and so forth. The protein on the right we've seen before. This is PKR. I show you this because it's got signs of selection from multiple viruses. PKR, if you remember, is activated by double-stranded RNA, phosphorylates EIF2 alpha, and shuts down translation. And I told you, unless viruses encode antagonists of this protein, they're not going to be around. And look how many viruses are influenced by this protein. Herpes viruses, flu, pox viruses, etc. So they encode proteins that interact with different domains of this PKR. Excuse me, on the left you can see the RNA binding domain has signs of positive selection by these different viral proteins. The protein kinase domain on the right has signs of positive selection by those red arrows. Again, different viral proteins are interacting with that. So the overall picture is at the bottom there. The host defense protein, in this case PKR, has different sites on its surface that are bound by different viral counter defense proteins. And we can see these change on each side. We can see the host side changing in the phylogeny of the animals we're looking at. We can see the virus side changing as well. And so that's how we can map the evolution of host proteins driven by viruses. So our makeup has incredibly been influenced by all the virus infections that we've had over the years. One more example of this. This is amazing. This is the transferrin receptor, TFR. And its evolution has been, in mice anyway, or in rodents, has been shaped by two different viruses. So as you know, transferrin receptor picks up transferrin, which is bound to iron, and pulls it into the cell, has an important function. Transferrin is a dimer. You can see on both sides the same molecule twice. There it is linked to the cell surface. This is the receptor for two different viruses, an arenavirus, an RNA virus, and mouse mammary tumor virus, a retrovirus. And the arenavirus binding site is in gray, and it's repeated on both sides, the dimer. And the MMTV binding site is in blue. And this is distinct from the transferrin binding site, which is down here. So because it's distinct, the protein can change to evade virus binding. And that's what these red spheres are. These are changes that we can pinpoint in the phylogeny of rodents that avoid binding of arenavirus or MMTV. And of course, the virus can then change back to be able to bind again. It goes back and forth in an arms race over and over. The point here is that this protein's been shaped by this arms race, and it can happen because the virus binding sites are away from the transferrin binding site. So you can see there's a lot of genome diversity that enables the ability to evade these kinds of host proteins that would block virus infection. Yet, when we look at viruses, say, serologically, in some cases there's a lot of variability, like you would predict from what I've just told you. But in some cases there's not. There are only 3 serotypes of poliovirus, but over 150 rhinoviruses. These are 2 viruses from the same family. Why is there such a difference in the diversity? There's only 1 measles serotype. We've been using the same measles vaccine for over 50 years, and the virus has not evolved to avoid it at all. The antibodies we make against measles, the measles virus does not evade them at all, whereas influenza viruses vary on a year-to-year basis. And there are many more examples of this. So despite all this flexibility and the ability to overcome cellular defense proteins, for example, as I've told you, some viruses have considerably less flexibility when it comes to antigenic variation. And why? The question is why? And I don't have an answer for you. It may rest in the architecture of viruses. Maybe the polio capsid is more constrained than the rhinovirus capsid. Maybe the measles glycoprotein is more constrained than the influenza glycoprotein. Or maybe it's the error rate. Maybe a small difference in error rate can accommodate this. So we don't know the answer to that, but it's a very striking observation that some viruses vary against the immune response quite a bit. That's what we're talking about here, and others very little. Here's another example of selection that I want to tell you about, and this involves virulence. This is a question I'm very interested in, and the question is virulence a positive or negative selection force? So if it's positive, you would say increased virulence is a consequence of high viral loads that you need for transmission. in order for viruses to be transmitted, you select for ones that replicate to high titers, and those are the best ones at being transmitted. And because you're replicating higher, then you cause more disease in the host. It's not that virulence itself is selected for, but a property that is required for transmission. If virulence selection were a negative trait, then the idea would be increased virulence reduces transmissibility because hosts die faster, and they don't have a chance to transmit the virus to someone else. So that's two ways of looking at whether virulence would be a positively or negatively selected trait. Unfortunately, the answer is both, because we know viruses that are of high virulence and of low virulence circulating today. And so it's not a simple one or the other. But I do want to tell you an experiment that illustrates that virulence can be a negative selection factor. And this starts with an experiment that was done in Australia. In 1959, the European rabbit was introduced to Australia for hunters because the native Australian rabbits were too fast and they couldn't shoot them. So the European rabbits were slower and they could shoot them. So they brought them in, and unfortunately there were no predators of these rabbits, and so they reproduced like crazy. You can see from this picture. They were plague. So they said, no, we have to get rid of these rabbits. Of course, they already opened the door. It's kind of late to do that, but they decided to use a virus to do that, a pox virus called myxomavirus. And it was released in the 1950s to try and get rid of the rabbits. Now, the natural host of this virus is the cottontail, which is not the rabbit they released. The virus is spread by mosquitoes, and these cottontails get warts on their ears, but they're fine. The European rabbits, infection is 90 to 99% fatal. So that's why they released this virus into the European rabbit population. So what happened? First year, the virus killed at a 99.8% mortality rate. But that 2.2%, man, that gets you. The second year, the mortality rate dropped to 30% because those 0.2% of rabbits had polymorphisms that allowed them to survive after virus infection. There's so many rabbits, the genetic diversity was so great that just a few could escape, and they reproduced the next season. Forget about eradication. They could never eradicate. These rabbits are still there in Australia. In fact, people have been studying them to see how their genome has changed, and the antiviral proteins changed in these rabbits were selected for so that the viruses can no longer kill them. It's a very interesting story. This is an example of an experiment in the evolution of virulence, because both of the sides of this equation, rabbits and viruses, make large numbers of offspring. The virus probably evolved to kill fewer rabbits, so they would overwinter and spread the virus in the spring by mosquitoes. So it's a natural evolution to less pathogenesis. The rabbits became more resistant, and that is one way you would predict an evolving pathogen would go, to kill just enough animals so that it can be transmitted to another host. You wouldn't want to kill all of them because then you have no more hosts. Now, just as a sort of coda to this story, more recently, they have a carp problem in Australia. You know, carp are farmed fish. So they have farms in Australia where they grow them, but they had a flood one year, and a lot of the fish got out into the local rivers and reproduced like crazy, and they want to get rid of them. So they're thinking about using a herpes virus here, as if they didn't learn a lesson from the rabbits. But as you can see, this may not work. If this did work, there would be millions of pounds of dead carp in the rivers that they would have to get rid of. That's one of the problems that they haven't dealt with. What about people, though? What can we say about viral evolution in people? Well, as we'll see next time, some recently transferred viruses to humans, like Lassa, Ebola virus, and HIV, when the virus first goes from an animal into human, it tends to be virulent. If you look at viruses that jumped a long time ago, thousands of years ago, polio and measles, although they can kill people today, they appear to be far less virulent. The question is, what happened in the meantime? Unfortunately, for human viruses, we don't know because we don't have original isolates. And so we can't answer this question. Back to viruses mutating. Nevertheless, we, meaning scientists and journalists, are obsessed with virulence. Here are some more quotes, Ebola virus is mutating and it will go airborne. That was a New York Times opinion. And this was the mutating Ebola virus, which I already told you about. Here's Zika. Peter Hotez says Zika probably mutated. It mutated from an African to a pandemic strain and then spread. It's easy to blame mutations. But as I'm going to tell you next time, for poliovirus, there are other explanations. And in all cases, it's impossible to show that mutation has done something to a human virus. We have no data on the evolution of virulence in humans because we don't have the original virus before it got into people. And even if we did, we couldn't learn anything because you can't infect people with it to compare its virulence with that of the viruses that are circulating today. We can learn a lot about evolution of virulence in animals, but not in people. So whenever you see a headline about viruses evolving in people, be very suspicious. Let's talk a little bit about the origin of viruses. You know, we can tell about the origin of many species here on the planet by getting fossils. For many years, we didn't have any very old virus specimens. Oldest for a long time was 1918 influenza virus, which was actually recovered from the sequence. And more recently, a giant virus shown here, Pithovirus sibericum, was recovered from permafrost in Siberia. It was about 30,000 years old, but that's not old. You can't tell very much about evolution from that. Fortunately, we have something even better. We have a fossil of sorts, and that is molecular clock analysis. So if you look at herpes virus genome evolution and compare it with its host's evolution, you can extrapolate that the herpes viruses arose about 180 to 220 million years ago. That would put it around the dinosaurs. So just think, dinosaurs had cold sores, just like us. They get a little stressed. T-Rex can't get a meal, gets stressed out, he gets a cold sore. Even better, though, is endogenous viruses. I've told you so far in this course that retroviruses integrate into the genome of the infected cell. But it turns out that for many other viruses, both RNA and DNA, random bits of the genomes are just accidentally incorporated into the chromosomal DNA. These appear not to have any function. They're just accidents, but we can use them as clocks. And here's how that works. Here we're looking at rabbits. And we have a phylogeny of rabbits that we can compare by using phylogenomics. And at some point here, a long time ago, in the evolution of this particular line of rabbits, a piece of viral DNA integrated into the genome. And we can see it by sequencing the genome of these rabbits. We can see, there's a piece of X virus DNA integrated right here. And if it's fixed in the animal, if it's fixed in the germline, it will now be in all subsequent animals that have been derived by evolution from this ancestor. And so these two species, A and B, both have the endogenous viral element, and you can see it's in the same place in the chromosomal DNA. So that means you can tell exactly when it was integrated by comparing species She, which is Eve negative. And so you can say, knowing the age of this phylogeny of the host, you can say roughly the maximum age and the minimum age of the Eves. So you can get an idea of when viruses were circulating. And here's a picture of that for a couple of DNA viruses, parvoviruses, dependoviruses, and circoviruses. And here's a phylogeny of a variety of animals here. We can map when these viruses inserted into the genome. Again, these are accidental insertions of viral DNA. Not functional at all like retroviral proviruses, but they give us the opportunity to say this virus was circulating 60 million years ago, or this virus was circulating 50 million years ago, because we know the phylogeny of the host. We have sequenced the genomes of all of these host animals. And so that's why in the first day of this course, I told you that some retroviruses were thought to originate over 450 million years ago. We can see them integrated into the genome of a variety of species that are consistent with that. The Ordovician is down here, age of invertebrates. But I do think that viruses probably originated even before that, probably billions of years ago, maybe even before there were cells. The idea that currently I like is that there was a time in the evolution of Earth a pre-cellular stage where the Earth was populated by what we call replicons, just self-replicating nucleic acids. And this started as an RNA world. These were RNA replicons initially, and then probably later on became DNA. At some point, cells emerged. The first cells we call protocells with compartmentalization. And these replicons, which previously are replicating outside of cells in just the organic environment of the early Earth, they went inside of these cells because that's a good environment for a replicon to grow in. And they picked up genes from the cell that encode their capsids. Most of the capsid proteins of viruses that we have today, we can see had some origin in a cellular gene. So the replicons stole capsid proteins from the cell, and they become true viruses. And there we have our modern cell and modern viruses all starting at this precellular stage. And sometimes these viruses became large and over time lost genes, or sometimes they started small and acquired genes as they evolved. All the human viruses that we have probably originated long before Homo sapiens appeared on Earth. All of the viruses that infected us evolved from some other animal virus, whether it's an ancestor of Homo sapiens, so some of these homos or Paranthropus or Kenyanthropus, some of these may have had viruses that passed them down to us, or we may have acquired them more contemporaneously from other animals as well. But we have no viruses that actually originated in us. Let me give you just two examples of this origination of human viruses from an animal. Smallpox virus. Phylogenetic analysis of all the viral genome sequences suggests they arose about 3,000 to 4,000 years ago in East Africa. It looks like they were transmitted from camels. Now, camels are not native to Africa. They came from North America 45 million years ago, and they crossed over the land bridge and made their way to Africa about 3,500 to 4,500 years ago. And when they got there, they were greeted with an ancestral pox virus, probably carried by a gerbil. And this hatched oval is the gerbil range in Central Africa. It coincides with where the camels came in. So we think the camel got the virus from a gerbil, and then it diversified extensively in the camel. And then the camels, of course, were domesticated by people for their use, and that most likely introduced smallpox into people. So it came from an animal. And measles virus. It's most closely related to rinderpest, which is a pathogen of cows. And we think measles evolved from an old rinderpest when we first began to domesticate cattle. We didn't always raise cattle for milk and meat. It only happened, in the 11th and 12th centuries. And we got their viruses. And in particular, this is an interesting virus because it needs a population of between 250,000 and 500,000 people to be transmitted. So it only arose when cities began to reach those populations, which happened in the Middle East around this time. And then the virus was established there in these large population centers, and then, of course, traveled around the world. As people began to travel, they took measles virus with it, and it reached the Americas in the 16th century. They had never seen it, and it destroyed Native Americans by being introduced because they had no immunity to it. So if I say all viruses in humans always come from animals, why couldn't they just arise de novo? Let's do an experiment that looks at that. What's the number of all possible mutations of a viral genome that would give rise to a new virus? Well, sequence comparisons have shown that over half of the nucleotides in an RNA genome can accommodate mutations. So let's make the assumption that that's correct. Of an RNA virus, over half of the bases can be mutated. So for a 10 kb viral genome, that means you could have 4 to the 5,000 different sequences arising during replication. And that would be even higher if you consider deletions, recombination, and reassortment. It's a huge number. This is the number of atoms in the visible universe. 4 to the 5,000 is huge. So why don't we see new viruses arising? Why do we always have to get them from animals? Well, but that's because fundamental properties of viruses are probably constrained. We can recognize herpes and influenza. Some kind of stability is clearly maintained. And that includes the fact that extreme alterations don't survive. So you could put lots of mutations in a genome, but it wouldn't be viable. And that probably constrains evolution. And there are many properties that constrain it. The viral genome, DNA can't become RNA or vice versa. We've never seen that happen. For replication, you need to interact with host proteins. So you can't have major changes in replication proteins. mRNA synthesis, RNA structure, all of this is constrained. So you can't have large shifts. And the physical nature of the capsid, of course, icosahedral capsids have a defined interior space. So it can't get huge, it can't get much smaller. And then, of course, selection during infection. If you're too efficient at bypassing defenses, you'll kill the host, you will never be transmitted. And if you don't replicate well enough, you're going to be eliminated. So we think that all of these constraints limit the number of new viruses that emerge, and that's why all of our viruses always come from another animal rather than emerging de novo in us. And I'll leave you with this thought. It took 8 million years for humans to diverge from chimpanzees. Today, our genome is 98% chimp. It took 8 million years for that nucleotide sequence to emerge. Poliovirus, in going from the mouth to being excreted in the feces, does the same amount of variation, 2%, in 5 days. So imagine what a virus could do with 8 million years. All right, next time we're going to take this information and put it to use and talk about how emerging viruses arise.